So today, I would like to start with this question. So yeah, COVID-19 is a big problem, but why are we threatened by the disease? So firstly, morbidity and mortality rates are relatively high. So in developed countries, including the UK, approximately 1% of all infected people are considered to die. And also, the virus is fast spreading. Infection rates are really high. And therefore, unless controlled, hospital admissions can be dramatically increased within a short time. So this is the major problem. And the reason why we have such dramatic temporal and spatial disturbance is that the vast majority of people do not have immunity. And of course, we are hoping that the vaccines will change the situation. And this is the virus of COVID-19, which is called SARS-CoV-2. And it has several major proteins, making a shell to include their genomic information, gene information inside the shell. And the viral genome, we call it genome, which means basically all the viral genes in SARS-CoV-2 is coded by 30,000 nucleotides, which are written by the 4 characters for A, U, G, or C. And we don't, so this shows the sequences, and it's written in this way, and this guy is threatening us. So today I'm not going to talk about the details, but I would like to highlight one protein called spike protein, which is a very important protein, viral protein for immunity and vaccination. And also it's important for virus to infect human cells. So SARS-CoV-2, the virus, binds human cells using the spike protein, which binds to the human protein called ACE2. And this protein is found in epithelial cells, in the mucosal cells, in the nose, the lung, the oral cavity, and in the intestines. So it's quite straightforward to understand the common symptoms of COVID-19, which include cough, pneumonia, and gastrointestinal symptoms, nausea, diarrhea, loss of smell, loss of taste. So these are due to the damage caused by the virus, which infects cells in those mucosal surfaces. So, but the virus binds ACE2 and goes into the human cell in order to replicate themselves, and eventually the viruses will destroy cells to go outside and will infect other cells. We have immunity to fight against the virus. Now, I would like to start with the most relevant cells in this topic. One of the most relevant cells, B cells. So B cells produce antibodies, which bind the virus. And some antibodies can bind the virus and block the infection. And such antibodies are called neutralizing antibodies. So we are hoping to produce such antibodies to protect us from infection and also to recover from the infection when we are infected. So by having such antibodies, the virus will no longer be able to enter the cell, human cells. We know that people recovered from COVID-19, convalescent patients, develop neutralizing antibodies against SARS-CoV-2. And those antibodies bind S protein. So this shows that as you go, as going to the rightwards, the dilution of the serum is increased, and there are 1, 2, 3, 4, 5 sera from 5 former patients. And then when the serum is not diluted, then they can completely block the infection in vitro. But if it's diluted, then of course the effect is diminished. But this way we can precisely determine the effects of each serum or each antibody on the virus and how much each can protect us from the infection. So using such experiments, scientists have established that it's really important, critical, to develop good neutralizing antibodies against the S protein. So vaccines, most of vaccines use S protein to allow B cells in people to produce antibodies, neutralizing antibodies against the S protein. So this is the result from Oxford vaccine during the clinical trial. And this is control, and this is the serum from people who were once vaccinated, and this is twice after the boost dose. And if you go up, then this means that the sera has a high concentration, high titer of antibodies that bind S protein. So the first dose can nicely upregulate such antibodies, and the prime dose further increase the antibodies by 10 or 20 or 30 times more than the prime dose. And then the antibodies will become equivalent to the ones in convalescent plasma samples, which means the sera from recovered patients. And convalescent serum is also an important therapeutic reagent. We can get blood from patients, convalescent patients, so people who recovered from the infection, and we can use the serum from the blood as a treatment to protect people who are vulnerable to the infection, so who have a high chance to develop a severe disease. So therefore, it was a natural strategy to develop therapeutic antibodies against S protein to be used in hospitals to prevent severe disease or to treat severe disease in intensive care unit. And such industry-produced monoclonal antibodies, we call them monoclonal antibodies, but these are safer than the blood product, and each monoclonal antibody binds to and recognizes only one part of S protein. And industry developed many such monoclonal antibodies to be used in hospitals. So in this talk, I will show some data related to the antibodies. So now, as we know, as you know, the vaccines have been successfully developed. And it was really a good news. So we didn't know how good any vaccines would be developed against the virus. And the results were just excellent, so exciting that we have become quite optimistic about the future of the pandemic. This is a result from Pfizer-BioNTech vaccine, and the vaccine protects 95% of people after the second dose. Even after the first dose, 50% is protected. And data suggests that the vaccine can prevent the development of severe disease. And the safety is well confirmed, and other two major vaccines, Moderna and AstraZeneca's ones, have also high efficacies equivalent to this Pfizer's one. So these are very good news. And at the same time, when we heard the news, we also learned that the variants were emerging. So now I would like to talk about how the virus can be mutated. And so this slide shows how the virus is reproduced, how the virus replicates themselves. Firstly, the virus binds a human cell through ACE2 to go into the cell to release their genome inside the cell so that the virus can exploit the machineries in the cell to replicate themselves. And firstly, the RNA inside the virus will be translated into viral proteins, and then the virus will use the proteins to replicate themselves. So for the genome to be proliferated, every creature needs to replicate the genome. And this is a sort of a transcript work. So each nucleotide needs to be precisely replicated so that identical things can be produced. Identical viruses can be produced. However, no system is perfect. So always there is a chance that this machinery will wrongly replicate the genome when mutations occur. But mostly they are fine, and the replicated genomes will be packed into viral proteins to be released outside to infect other cells. So this is the life cycle of the virus. And for your information, some antiviral drugs like remdesivir will block this replication process. So this is a major process for the virus. And the virus has a proofreading function, and therefore it acquires mutations only at relatively low rates. Only one base, only one base per a million replication of each nucleotides. So 10 to the minus 6 per base per replication. On the other hand, the surveillance data for viral mutations worldwide show that the virus acquires 1 to 2 mutations per month in all regions in the world. So the virus here randomly accumulates different mutations in different locations. However, there are many factors. The virus randomly accumulates mutations, but the spread of each mutated virus may not occur at the same rate. So and we need to look into a lot of data to understand their dynamics. So today I would like to show you a very basic principle of viral mutation and selection here. So the virus may acquire mutations when replicating themselves itself. And most mutations do not change any property. So they are still the same, exactly the same virus. Some mutations may rather inhibit their function. Then the virus will become less functional and will be gone. So this is a good thing. However, when viruses get such random mutations, occasionally some mutation may have a biological significance. So in this schematic figure, so all viruses look the same apart from this guy, which lacks a little small part of the S protein. And in this situation, this mutant with a meaningless, this mutant the mutation doesn't have any significance. So this is meaningless. However, in some situations, for example, when an antibody is produced in this environment, in this person, then assuming that this antibody binds this yellow bit, this triangle bit of S protein, then this binds this virus and this no longer infects other cells. Then they will disappear. And many antibodies are produced by B cells and all will disappear apart from this mutated virus. And this cannot be recognized by the antibody. Then the virus can escape the immune response and can proliferate happily in the body. So this way, immune escape mutant can become dominant and the same antibody no longer works. So this is, we call it antibody pressure or immune pressure. Under such a pressure, immune escaping viruses can be selected from randomly mutated viruses. So this is the mechanism for how random mutations can lead to a biologically significant mutation. And this is the UK variant B117 or VOC 2020-1201. So this variant has 17 mutations in its protein, including 8 mutations in S protein. So this is quite a lot. And I'll show you two mutations in the next slide, but it has such a mutation, N501Y, which means that the amino acid, the number 501, is changed from N to Y. And the deletion of 69 to 90 amino acids from the S protein. And this variant is estimated to have approximately 30% higher transmission rate. And it, as we know, it spread fast in Southeast England and London in December and replaced the epidemic in those regions. So this shows East England, London, and the Southeast. Approximately from December, the percentage of the virus detected in those areas show that the mutant, the variant, became dominant in those areas. And scientists speculate that such variants may be able to efficiently evolve in immunocompromised patients because of several case analysis. So the first analysis, in this analysis, an immunocompromised patient who does not have a functional immune responses much was infected by the virus. And then, so the patient was treated by convalescent serum or monoclonal antibodies throughout 150 days. And during that time, the researchers collected samples and analyzed the genome of each viral sample and found that gradually the virus accumulated new mutations in the patient, and the virus became quite different from other viruses circulating in the area. Notably, it acquired N501Y, which is found in all the major variants we have right now. Another patient, so this is another immunocompromised patient, and in the patient there were slightly different viral strains, and sometimes some viral strain dominated, and at some other time points, other viral strains dominated. But eventually, during the clinical course of 100 days, the virus acquired a lot of different mutations, including DEL 69 and 90, 69 to 90, which is found in the UK variant. So it seems that the variant, the UK variant, is most likely accumulated multiple mutations in such a patient, and thus appeared as a very significantly different variant when scientists detected the virus. And we are still on our way to understand the meaning of each mutation, but N501 and DEL 69 to 90 may have some functional significance. Next, this is the South Africa variant, 501Y.V2. As you may know, South Africa suppressed the first wave by October 2020. So the peak was around July, August. And after the successful suppression of the epidemic, cases increased again in Nelson Mandela Bay area, and since November, and the second wave was even bigger. And notably, the second wave was caused by the variant, South Africa variant, which has 21 mutations, and it replaced the epidemic in the area. And in these 21 mutations, 9 mutations are found in S protein. So this is S protein, and this bit, this part binds to ACE2. And the South Africa variant has 3 mutations in this critical part, which include N501Y, E484K, K417N. it's like a code, but there are such mutations. The concerning thing about the variant is that the virus seems to be resistant to multiple therapeutic monoclonal antibodies. And the graphs show that if the points are found up higher, had higher values, it means that the neutralizing activities are high, or S protein binding is high, very good. The original strain binds to the monoclonal antibodies. There are 3 antibodies shown here. So the original strain binds nicely to all the 3 antibodies, but the South Africa variant doesn't, and it's not well neutralized by the 3 antibodies. There are many other monoclonal antibodies, so some are effective, but it seems that the variant acquired such mutations. And another thing here is that the variant is resistant to convalescent sera from people who contracted the virus, the infection, during the first wave. So this means that the antibodies that are produced by people by the infection by the original strain of the virus may not be always effective against the variant. And in this preliminary study, so the scientists show that the original virus was nicely blocked, suppressed by such convalescent sera from the first wave, but the South Africa variant is not completely suppressed. Among 44 sera, 44 people who donated convalescent serum, about a half of such sera did not have good antibodies to block the infection. So the half of those sera could not neutralize the virus. Only using the 3 mutations in the part of the S protein that binds to S2, the reduction is quite significant in this study. The yellow, so this part is the percentage of the sera that was not effective to neutralize the virus. So next, the slide shows the Brazil experience. In the area, Amazonas area in Brazil, in the capital, Manaus, they had a huge first wave in the spring, peaking around May. And they did lockdown and physical distancing to control the infection, but scientists analyzed the area, the population, and estimate that about 76% of the population was infected by October 2020. So some people claimed that the people in Manaus may have established herd immunity. However, since November, the infection gradually increased, and December, the growth became exponential when scientists detected a new variant called P1. And the second wave was even bigger. So what does this mean? So firstly, at least by natural infection, herd immunity may not be established. if the estimation by the scientist about the rate of infection is correct. Possibly this is because immunity decays within a year, or possibly because a big wave will produce immune escape variants. And this is the variant P1. The left figure shows the similarities, the relationships between sorry, sorry, can you, I'm sorry.
Sorry, this is for your printer.
Yeah.
Okay, sorry.
I'm sorry for this disturbance. Yes. Yes, the left figure shows many viruses here, and their genome sequences are compared, and the similarities are shown by the distance. So there are many, many viruses circulating in Brazil, and the closer the lines are, the more similar the viruses are. And here, the viruses isolated in Manaus were very different from other viruses circulating Brazil. And they found that there are quite a few mutations, 21 mutations acquired by the variant, which include the immune escape mutations E484K and K417T. So let's see, let's look into why we say those mutations immune escape. This is an experiment in vitro. The scientists cultured the virus with convalescent patient's serum for quite a long time, about for 100 days. Then the virus replicates itself in the culture in the presence of antibodies in the serum. Then what happens is about 40 days after the culture, the virus started to show some mutations. Sorry. And about 80 days after the culture, the virus acquired the mutation E484K and also other mutations. And as the virus acquired mutations, the virus became more difficult to be neutralized by the convalescent serum used for the culture. So this means that in vitro, we can induce such an escape mutant that is resistant to the antibodies incubated with the virus. And this E484K is found in the South Africa and the Brazil variants. I would like to show this very much interesting experiments. Do not worry, I'm not going to talk about all the text in this slide, but this is more simpler than imagined. So there are two experiments. In the first experiments, the scientists tested various mutations, various mutants for their sensitivities to therapeutic antibodies. I told you that there are many monoclonal antibodies to block the virus, and now they are classified into class 1, 2, 3, depending on which part of S protein is recognized by the antibody. So then, so this is the first experiment. The second experiment is, in this experiment, researchers incubated the virus with each antibody. Okay, so then let's see one example. So please look at these rectangles. Then in the first experiment, the mutant K417N is resistant to monoclonal antibodies, particularly class 1 and class 1-2 antibodies. So the antibodies bind some parts close to the S2 binding part. Okay. In the second experiment, by incubating the virus with class 1 and class 1-2 antibodies, the virus, a lot of viruses acquired the mutation K417 something. Okay. In this example, the mutant E484K is resistant to class 1-2 and class 2 antibodies. And when the SARS-CoV-2, the original strain, is incubated with the class 2 antibodies, then the virus acquired the mutation E484K. So does this make sense? So there are some close relationships between which parts of S protein are recognized by monoclonal antibodies and which mutations could be induced. So the virus can evolve to escape from each antibody if it is incubated for a long time with the antibody. And N501Y is quite similar. So this is resistant to class 1, 2, 3, 3, and this mutation can be induced in vitro. And we know that N501Y is common to all the variants, and K417, E484 are found in the South Africa and Brazil mutants variants. And we know that a small number of the UK variant has acquired E484K recently. So now the topic is about the vaccines. So are vaccines effective against the variant? And this is a study, recent study, analyzing the neutralization antibody titer, comparing the original strain, the UK variants and the South Africa variants, vaccines from Moderna and Pfizer. And then the UK variant has approximately 2 times reduced sensitivity to the antibodies that are produced by the vaccines. And South Africa variant is about between 6 to 8 times less sensitive. And please note that this is the neutralization antibody titer. So this doesn't mean that the protection is halved or anything. We rather think that this is a minor reduction, particularly UK variant is well neutralized by those antibodies. South African variant is slightly less sensitive. So we need to wait for the clinical trial results. But this is the table showing some updated figures for the efficacy of each vaccine to circulating viruses in each area, each country. So we know that Novavax is recently developed. So it has latest figures. The efficacy of Novavax vaccine is very good in the UK, but is reduced slightly in South Africa. And this is a new class of vaccine, but still uses the same protein, S protein. Johnson & Johnson shows such figures. The vaccine is effective at 72% to the original strain, while the efficacy is slightly reduced to 57% in South Africa. We don't know we do not have data for the Brazil variant. So we would need to wait for the results, but people expect that the mRNA vaccines should have an enough efficacies against the major variants. So let's see. This is a summary of this part. The virus can acquire escape mutations under antibody pressure, and the South Africa variant is considered to have emerged by acquiring such escape mutations through infecting many people, or possibly through immunocompromised patients too. The effects of some vaccines on the South Africa variant are slightly reduced, and we don't have data for the Brazil. So in the last part, can I ask you how long do I have still? I can't see the.
It's about 1:45 now, so yeah, but you can have another 5 minutes. That's fine.
Yeah. So then, very quickly, I would like to show T-cell story. So because people expect that T-cell immunity must be more robust than antibodies. So eventually, if we acquire T-cell immunity through vaccines, hopefully, sometimes through infection, the immune system should more accommodate the situation. So this is, however, the outstanding issues include how long is T-cell immunity maintained, and does the virus escape from T-cell immunity too? And does T-cell immunity suppress the development of a severe disease? So very quickly then, T-cells do not see directly the virus. So T-cells can see the fragment of viral proteins which are presented by human cells. So in this case, the infected cells are eaten by a professional immune cell, which show a part of a fragment of viral proteins to T cells. And then, so the T cell can see a very small fragment of the viral protein and start initiate reactions. There are two different types of T cells. One is called helper T cells because they can help other immune cells, particularly B cells, and the other type of T cells, killer cells. And they organize, coordinate immune responses, so they are sort of a conductor. And this cell is critical for protecting us from viral infections. In addition, another type of T cells is called killer cells, which can directly kill virus-infected cells by recognizing a fragment of viral proteins, which is presented by infected cells. So there are such cells, but both T cells recognize the very tiny part of viral proteins. And this study analyzed precisely which part of S protein is recognized by either antibody or T cells. Then very interestingly, they found that maybe this is intuitively understandable, but the antibodies bind to the surface. So they bind spike protein on its surface, but T cells recognize more often the internal part of S protein. So red parts are recognized by antibodies, and the yellow parts are recognized by T cells. So this is one of the reasons why scientists expect that T-cell immunity may be more robust than antibodies because viral proteins can change the shape of the surface quite easily, but it should be more difficult for the virus to drastically change the inner structure. So very briefly, this is about the longevity of the immunological immunity. So researchers analyzed how long memory B cells and memory T cells are maintained after the infection. And then they found that B cells are quite stable. Antibodies can decay into the half within 140 days. But still, it's quite stable, isn't it? And especially memory B cells are very stable over 8 months after infection. T cells are okay. CD8 T cells decay to the half in 125 days, and the CD4 about 94 days. So T cells decay more quickly, but we also know that once recognizing antigens, T cells can duplicate themselves within a day. So the reduction seems okay. So the immunological memory should be maintained quite well. So, but we still don't know what types of T cell activities are protective against COVID, especially protective against severe disease. Because T cells can sometimes do the negative things too. So we still don't know. So what types of T cell responses are protective against severe disease? And I'm going to show some aspects of T cells from using a couple of slides from this slide. Very simply speaking, T cells are important, critical to protect us from viral infections. So they induce protective response, but at the same time, they induce damages to tissues too. So when they are overreacted, then they may induce organ problems. So this is because T cells are always seeing the self tissues as well while reacting to foreign invaders. So this is, again, very briefly, as you may have heard, severe patients may develop a situation, a condition called cytokine storm, in which patients develop a lot of proteins called cytokines, which are produced by immune cells. And cytokines activate other immune cells and blood vessel cells and other part of cells in the body. So cytokines can too much activate various systems, and this may lead to many problems, including circulation problems as well. And researchers identified that many different cytokines are more produced by severe patients compared to moderate patients or healthy people. And there are 1, 2, 3, 4, 5 different cytokines shown, and this suggests that a wide range of immune cell populations are most likely very much activated, and something is happening. Talking about T cells, T cells show one of the most remarkable features of severe patients, and they are remarkably reduced in severe patients compared to moderate patients and healthy people. This is the total percentage of T cells, total number of T cells, the helper cells, CD4 T cells, and the killer cells, CD8 cells. They are all very much reduced in severe patients by some reason. We don't know the reason yet. We speculate that many T cells in severe patients may be too much activated and dying quickly. Or some people say that activated T cells may migrate into tissues and disappear from the blood. We are still on the way to understand what is happening in severe patients, but we investigated severe COVID and moderate COVID and their lung samples data and found that in severe patients in the lung with pneumonia, there are a lot of very much activated CD4 T cells. So helper T cells are very much activated. At the same time, we also found that a very important brake molecule that inhibits T cell activities is very much reduced in severe patients in their lung. So this is called FOXP3. But we know that when FOXP3 is induced, as in moderate patients, the molecule can block T cell activities to prevent overreactions. So it seems that in severe patients, T cells are just very much activated, hyperactivated. And we think that probably T cells overreact to the infection and prematurely die and consuming many resources in the body, leading to severe disease. So this is just a hypothesis. Not yet connected to the variant story, but I wanted to introduce this at the end in order to show you the gap that we still have to address. So this is a summary, but probably now the time is quite too much. So I'm going to say the final word. The virus is mutating, but immunity and the technologies are evolving. So we don't have to be pessimistic. Probably we can be slightly optimistic while carefully observing situations to prepare fast reactions. And this is acknowledgement. Thank you for your attention.